Hey everyone, Dr. D here, and in this video we are doing Exam 2 review from our Talaro's Foundations in Microbiology 12th edition textbook by Chess. So let's go ahead and get started.
Dr. D, Dr. D, Dr. D. Dr. D, Dr. D, Dr. D. Dr. D, Dr. D, Dr. D. Explain stuff.
Alright, remember this exam will cover chapters 7, 8, 11, and 12. So let's get started with chapter 7. Please remember all of the essential nutrients we talked about, the difference between macronutrients and micronutrients. Specifically, you should remember chomps. Remember chomps. These are the big six elements that make up most of our cells' dry weight, right? We got carbon, hydrogen, oxygen, phosphorus, sulfur, and nitrogen. Please remember these six elements and how vital they are for life. And do you remember that organisms need a source of all of those elements? You don't have to memorize everything on this slide, but I would like you to know that some organisms can get their CO2 through carbon fixation. That means taking CO2 out of the air. Other organisms get it through their diet by consuming organic compounds. Same thing with nitrogen. Do you remember that some organisms can take nitrogen out of the air? And those are called nitrogen fixers, right? They do nitrogen fixation. These organisms take nitrogen straight out of the air, but there are other sources of nitrogen as well. Do you remember these nutritional types? Please remember all of these nutritional types, a heterotroph, an autotroph. Remember this table here? Please remember this table. For instance, where do organisms get their carbon from? Are they autotrophs or heterotrophs? Where do organisms get their energy from? Are they phototrophs or chemotrophs? Where do organisms get their electrons from? Are they lithotrophs or organotrophs? So I would want to definitely know the definitions of all of these terms right here. And you should know that plants, remember plants are an example of autotrophs, whereas animals like you and me and animal-like microorganisms are actually chemoorganoheterotrophs. That's something interesting to know. You should know that saprobes are decomposers. And by the way, do you remember when I said that these concept check questions can appear on exams? So make sure you learn all of your concept check questions because I could include them on the exam. This one states, if an organism is degrading large organic molecules to get both carbon and energy, it would be described as a chemoorganotroph. Remember, so please know those terms. You should know about transport. Remember, there's passive transport and active transport. Do you remember that diffusion, osmosis, facilitated diffusion, these are all passive processes. These are all passive transport. And do you remember that osmosis is the diffusion of water across a membrane? Do you remember that facilitated diffusion it requires solutes to cross through a carrier or transport protein? And do you remember with active transport, this requires energy? And the example we covered was bulk transport, endocytosis and exocytosis. Do you remember that water moves from where water is high in concentration to where water is lower in concentration? Do you remember what happens in isotonic solutions, hypotonic solutions, and hypertonic solutions? You should know that in hypotonic solutions, water rushes in. In hypertonic solutions, water rushes out. You should know that in a hypertonic solution, a cell that has a cell wall becomes plasmolyzed. However, in a hypertonic solution, A cell without a cell wall becomes shriveled or crenated. You should know that in a hypotonic solution, the, the cell with the cell wall becomes turgid or high pressured, whereas whereas in a hypotonic solution, the cell without a cell wall becomes lysed. Remember these tonicity questions here. And remember, simple diffusion, facilitated diffusion, osmosis, all of these are passive transport examples. Do you remember the difference between phagocytosis and pinocytosis? One involves ingesting solids and the other one liquids. And that leads to concept check number 3. If a cell is in concentrated glucose solution and the glucose is moving into the cell through a carrier protein, this would be an example of which of the following: facilitated diffusion, right? Because the solute is moving through a carrier protein or a transport protein. Remember the cardinal temperatures: minimum, maximum, and optimum. And by the way, please know these organisms based on their temperature requirements or adaptations, right? You got psychrophiles, which grow in the cold, psychotrophs, which grow in about room temperature, mesophiles that grow at body temperature, thermophiles, and extreme thermophiles that grow in hot conditions. Now, can you beat Wicket? Which one of these did we discuss would be most likely to cause a human disease? Can you beat Wicket? That's right, Wicket. Mesophiles, because they grow optimally at body temperature of 37 degrees C, right? Also, do you remember that it is an adaptation to be able to grow in the presence of oxygen? Not all cells can grow in the presence of oxygen, and that is because when cells grow in the presence of oxygen, they form these radical oxygen species or ROS. And unless those bacteria have the enzymes superoxide dismutase or catalase to mitigate the damage of ROS, then they cannot grow in the presence of ROS. Do you remember the different oxygen requirement types? Obligate aerobes, facultative anaerobes, microaerophiles, obligate anaerobes, aerotolerant anaerobes. Just know the difference between those oxygen types. Also with pH effects, neutrophiles, acidophiles, alkalinophiles, and where they prefer to grow. Halophiles, right? Remember, halophiles can grow in high salt conditions. Remember concept check number 5, Chlamydomonas nivalis grows in Alaskan glaciers, and its photosynthetic pigments give the snow a red crust. This organism would be best described as a psychrophile, right? Because those grow in the cold, in the Alaskan cold. Do you remember your symbiotic types? You should know that mutualism is a type of symbiosis where both members benefit. You should know that commensalism is where the commensal organism benefits. The other member is neither harmed nor benefited. And then in parasitism, the parasite benefits and the host becomes harmed. And then do you remember syntropy? In syntropy, here the members cooperate and share nutrients. Remember that. And then do you remember in quorum sensing? Quorum sensing is found in biofilm formation. Quorum sensing is a way for microbes to determine the number of microbes in their vicinity. Also, do you remember binary fission, how that works? The nucleoid becomes replicated in an asexual fashion. Those two copies of the chromosome, the circularized chromosome, move to opposite ends of the cell. The cell begins to split in half with the formation of what's known as a septum, and that results in 2 genetically identical cells. And you should know this equation for the rate of population growth, right? Because, there might be a question like this regarding, how many cells you would have at the end of a particular span of time based on a doubling time. And then lastly, do you remember growth curve in a batch culture, lag phase, exponential phase, stationary phase, and death phase? What's happening during each, and why does each occur? And lastly, do you remember turbidometry was used in order to measure the cloudiness or turbidity of bacteria growing in broth in order to determine the number of bacteria in that broth? All right, and that's it for chapter 7. A pretty short and sweet chapter with some good concepts on there to understand. And I hope I narrowed it down for you so that you know kind of what to focus on, in order to be successful. So let's move on to the next chapter, chapter 8. All right, chapter 8, let's do it. An introduction to microbial metabolism. You should know that metabolism includes all of the chemical processes that happen inside of a cell. It's basically everything that happens inside of a cell that requires energy. And then you have catabolism and anabolism. Remember the difference between the two? In catabolism, there are molecules being broken down to release energy, and during anabolism, there are molecules being built requiring the input of energy. You should know that enzymes are biological catalysts that lower the activation energy, thereby speeding up or catalyzing a reaction. You should know that enzymes aren't all necessarily completely made up of protein. Some are made up of RNA. Some require a cofactor. So there's an apoenzyme plus a cofactor. You should definitely know that these cofactors can be inorganic, which include usually metal ions like iron, copper, or magnesium. But then there can also be organic cofactors as well. And you should know that those are called coenzymes, and they include things like the vitamins. You should know that proper folding of an enzyme is important. Otherwise, if a protein becomes unfolded, that means denatured, it loses function. You should know that exoenzymes function outside of the cell, while endoenzymes function inside of the cell. Do you remember how synthesis reactions occur? Macromolecules are produced by a process known as condensation reaction, or by dehydration reaction or dehydration synthesis. Those all mean the same thing, essentially removing water to make a bond, while macromolecules are broken down by hydrolysis, introducing water to break a bond. Concept check number 1. Which of the following statements about enzymes is correct? They show no substrate specificity. That's false because they show high level of specificity. They are consumed by the reaction. No, enzymes are not consumed by the reaction they catalyze. They may be active extracellularly. That's true because remember exoenzymes. They are composed only of protein. That's false because many of them are either made of RNA or they require a cofactor as well. Remember that during competitive inhibition, the inhibitor binds to the active site, while in non-competitive inhibition, the inhibitor binds somewhere other than the active site. Do you remember redox reactions? Remember Leo says Gur? Loss of electron means oxidized. Gain of electron means reduced. So concept check number 3, if a molecule has been reduced during a reaction, it has done what? Well, remember reduced, Gur, gain of electron, right? So it has gained an electron. D would be your answer there. Now, you should definitely know the difference between aerobic respiration, anaerobic respiration, and fermentation. And one thing you should know is that these are all separate and distinct things. Aerobic respiration and anaerobic respiration involve glycolysis, the Krebs cycle, and that respiratory chain. You know what they mean by the respiratory chain? It means oxidative phosphorylation. It means electron transport chain plus ATP synthase with chemiosmosis. So remember the whole process of cellular respiration, aerobic respiration and anaerobic respiration both go through the entire process of cellular respiration, all the way from glycolysis to Krebs cycle to oxidative phosphorylation / respiratory chain. The only difference is that you should know this, The only difference between aerobic respiration and anaerobic respiration is simply the identity of the terminal electron acceptor, which picks up the electrons where? Can you beat Wicket? That's right, at the end of the electron transport chain. The end of the electron transport chain. For us aerobic respirators, oxygen picks up the electrons at the end of the electron transport chain, whereas for anaerobic respirators, a molecule other than oxygen, such as sulfate or nitrate or something of the sort, picks up the electrons at the end of the electron transport chain. And in anaerobic respiration, even though they don't make as many ATP as we do as aerobic respirators, they still make plenty of ATP. Now, you should know that fermentation is different than the other two above, and that's because in fermentation, this is when, let's say we run low on oxygen. We run low on oxygen because we're hitting the gym too hard or we're being deprived of oxygen. This means that all of those steps like Krebs cycle and the respiratory chain, oxidative phosphorylation shut down. And what's the only process of cellular respiration that keeps chugging along? Do you know? Can you beat Wicket? That's right. Glycolysis, Wicket. So yeah, glycolysis is the only process that's going on during fermentation. And so that means you're netting how many ATP, Wicket? That's right, Wicket, as always. 2 net ATP during fermentation, only 2 ATP, whereas aerobic respiration would net 38, according to your textbook, and anaerobic respiration would not quite reach 38, but it would be a lot more than 2 net ATP. Does that make sense? And do you remember during fermentation, this is where organic compounds are used as the final electron acceptor, such as pyruvic acid, pyruvate. And again, see the 3 processes juxtaposed here? The only big difference between aerobic respiration and anaerobic respiration is who's picking up the electrons at the end of that electron transport chain. For us, it's oxygen. For anaerobic respirators, it's sulfate or nitrate or carbonate, something like that. Whereas with fermentation, look, none of this stuff happens. The Krebs cycle doesn't happen. The respiratory chain doesn't happen. The only thing that actually is going on is glycolysis, and that results in production of fermentation products. And let me see if you can beat Wicket. Do all creatures produce the same fermentation products like us, like lactic acid? That's right, Wicket. No, different creatures, different microorganisms produce different fermentation products. You and me produce lactic acid during fermentation, when we hit the gym too hard, right? But other organisms like yeasts might produce ethanol, and other organisms produce a host of other chemicals such as butyric acid, ethanol, carbon dioxide, acetic acid, 2,3-butanediol, propionic acid. So you see, different organisms, you don't need to memorize all these, but you should know that different organisms produce different fermentation products. And fermentation is not the same thing as anaerobic respiration. And do you remember that you need to know the products of glycolysis, Krebs cycle, and that respiratory chain? So the products of glycolysis, you might want to make a note. The products of glycolysis, remember, are NADH, ATP, and pyruvic acid, aka pyruvate, right? Your textbook calls it pyruvic acid. Many textbooks call it pyruvate. It's the same thing. And what were the products of the Krebs cycle? Remember the Krebs cycle? The products are NADH, FADH2, CO2, ATP, right? And I believe that was it. Okay, so those are the products of the Krebs cycle. And then the products of the electron transport chain, the respiratory chain, or AKA oxidative phosphorylation at the end. Remember, the products there are water, because oxygen picks up the electrons and becomes reduced to water. So water and ATP. And then you should know that the difference between bacteria and eukaryotes in terms of where the electron transport chain is. Remember, in eukaryotes, the electron transport chain is in the mitochondria, and it's in those twisty, turny, convoluted cristae. Remember the inner membrane. The inner membrane of the mitochondria is where you find the electron transport chain. And so the protons are pumped into the intermembrane space. Whereas in prokaryotes, right, in bacteria, the electron transport chain is in the actual cell membrane, the plasma membrane, and the protons are getting pumped out of the cell. So isn't that neat? You should also know the overall equation for cellular respiration. Do you recall? Glucose plus oxygen form CO2, water, and ATP. All right, concept check number 5. Which part of aerobic respiration releases CO2? Remember, not glycolysis, because CO2 is not a product of glycolysis. It is the Krebs cycle. So B would be the correct answer there. And again, you should know that fermentation occurs when we run low on terminal electron acceptor, when we run low on oxygen. You should know that only glycolysis is occurring, which means we're only netting 2 ATP. And you should know that we also produce a fermentation product, during this process, because we are going to use an organic molecule such as pyruvate as our terminal electron acceptor. And then photosynthesis, you should know the equation for photosynthesis. Remember that CO2 plus water and light are the reactants, and the products are sugar and oxygen. You should know that there are photosynthetic bacteria as well as photosynthetic eukaryotes, which include, the algae as well as the plants and trees. And all of them do the light-dependent reactions, the light-independent reactions. You should know that in eukaryotes, photosynthesis occurs in chloroplasts, whereas in prokaryotes, it happens inside of the cell. Because remember, prokaryotes like bacteria, cyanobacteria, photosynthetic bacteria don't have chloroplasts. Chloroplasts are membrane-bound organelles. And you should know that light is captured by these photosynthetic pigments like chlorophyll. And that leads us to concept check number 7. The main job of the chloroplast pigments during photosynthesis is to capture photons of light. That's A. All right, that leads to the end of chapter 8. Let's do our break time with Gizmo and Wicket, come back strong, and finish off this exam review so we're prepared for exam 2. Let's do it. All right, welcome back from break time with Gizmo and Wicket. Let's carry on with chapter 11. Please know these terms in detail: sterilization, disinfection, antisepsis, sanitization, degermation, because I might ask for, the definitions of each. And remember, the gold standard is sterilization in terms of complete removal or destruction of all viable microorganisms and infectious agents. Remember that antisepsis is when chemicals are applied to body surfaces and tissues to destroy or inhibit vegetative pathogens. Germs with the highest resistance are prions as well as bacterial endospores. And the moderate resistance are exhibited by protozoan cysts, naked viruses, mycobacterium, and the least resistant are fungal spores, hyphae, yeasts, enveloped viruses, the trophozoites of protozoans. You should know that surfactants work by disrupting cell membranes. Here's concept check number 1. If you wipe your hands with an antimicrobial cleaner, you are accomplishing antisepsis, right? Because it's on living tissues. Remember the difference between moist heat and dry heat? At the same temperature, moist heat is a lot more effective. Also, do you remember the difference between the thermal death time versus the thermal death point? The thermal death time is the shortest length of time required to kill the microbes at a particular temperature, whereas the thermal death point is the lowest temperature required to kill all of those sample in 10 minutes. Remember, the autoclave is essentially like a pressure cooker or a pressure steamer. It achieves 15 psi, 121 degrees C for about 15-20 minutes to sterilize. Items are sterilized in an autoclave. However, you should 100% know that boiling water only achieves disinfection. You cannot sterilize using boiling water. Pasteurization is another type of moist heat control. Remember, this one is even lower temperature than boiling water in order to preserve the flavor of foods and beverages. Oh, you should remember this, that cold and desiccation do not kill microorganisms. Instead, they are microbiostatic. They just prevent them from growing. So cold and drying desiccation are not great ways of destroying microorganisms. So concept check number 3, which of the following accomplishes microbicidal effects? Boiling, yes. Pasteurization, yes. Incineration, yes. But remember, freezing and desiccation, that's more microbiostatic, right? So A, B, and C. Remember, we also talked about radiation or irradiation. And I told you that ionizing radiation, you should know that it includes gamma rays, X rays, and cathode rays, whereas non ionizing radiation includes UV rays. So you should know that. And you should know that ionizing radiation actually breaks DNA apart with double strand DNA breaks, while UV radiation, which is a non ionizing radiation, results in those pyrimidine dimers. Remember, the halogens include chlorine, bromine, iodine, fluorine. You should know how bleach works. Remember the hypochlorites or chlorine bleach? You should know that it is unstable in sunlight. You should know that it can be used to treat water, right? These are important things about chlorine bleach to remember. Iodine is important. It can be used as an antiseptic or a disinfectant. Remember, phenol is also a nice disinfectant, though it is somewhat toxic to us, our tissues. Concept check number 5 time. Which of the following is not true about hypochlorites or chlorine? Kills by denaturing proteins. Yes, it does. It is light sensitive. Yep. It is often used for disinfecting water. Correct. It slows the release of iodine. That one it does not do. Remember, alcohols at 70% are the best, most effective at 70% concentration. Hydrogen peroxide, you should know that this one produces highly reactive ROS, right? Reactive oxygen radicals. And it also releases oxygen gas during the process. You should know that the aldehydes are important. Glutaraldehyde at 2% is used as a sterilant in the clinic. Formaldehyde is usually used as a preservative. Sterilizing gases are important for sterilizing equipment that's heat sensitive, such as those depicted here. And again, concept check number 7, which of the following kills by producing oxygen radicals? Remember, we just talked about hydrogen peroxide doing that. Remember, heavy metals are oligodynamic, meaning they are effective at very small concentrations. All right, and that's it for chapter 11. I tried to touch on those important points there. Let's move on to our final chapter for this exam so that we're prepared. All right, this is it, chapter 12. Let's do it. Chapter 12, we're talking about antimicrobial chemotherapy. Remember that the perfect drug does not exist. Antibiotics are treatments against bacteria. You should know that some are natural. Many bacteria in the genuses of Streptomyces and bacillus produce antibiotics. Many molds, which are fungi in the genre of Penicillium and Cephalosporium, they also produce several antibiotics as well. Please know all of the terms on this list for the exam. Also remember that these are the 5 targets of drug action. You should know that penicillins and cephalosporins attack the cell wall. They block synthesis of peptidoglycan. You should know that polymyxins attack and disrupt the cell membrane. You should know that chloroquine and quinolones disrupt and affect nucleic acid synthesis. And it's the aminoglycosides, including streptomycin and gentamicin, that block protein synthesis. And do you remember those drugs that block protein synthesis usually bind to and inhibit the ribosome? See? And then you should know that the sulfonamides, remember the sulfa drugs, they are the ones that affect metabolic pathways, right? In fact, what they do specifically is block the metabolic pathway for production of folic acid. So again, concept check number 3, aminoglycosides are antibacterial drugs that target what? Remember, it's protein synthesis, C. You should know that the penicillins contain a beta-lactam ring. And do you remember the structure of the penicillins? They have the thiazolidine ring, the beta-lactam ring, and they also have another side chain. They kind of look like these. And do you remember, can you beat Wicket? There is a enzyme that penicillin-resistant bacteria can make, and it's called penicillinase. And by having penicillinase, these bacteria become resistant to penicillin. Why is that? What does penicillinase attack? What part of penicillin does penicillinase attack? Can you beat Wicket? That's right, Wicket. It's that beta-lactam ring. Remember the ring that kind of looks like a square here, made up of 3 carbons and a nitrogen. So penicillinase is an enzyme that penicillin-resistant bacteria form that actually cleaves this beta-lactam ring. Okay, so you should probably make a note of that. And do you remember that tetracycline was also an antibiotic that was important? Tetracycline blocks protein synthesis, though it can have adverse side effects. And so concept check number 5, which of the following drugs inhibits bacteria by blocking folic acid synthesis? Remember, that was the sulfa drugs or the sulfonamides. Next, antifungal drugs. Remember what I said, that antifungal drugs tend to have a low therapeutic index. That means that many antifungal drugs also have side effects on your cells as well and your tissues. And this is because why? You should know that this is because it's hard to treat fungal infections because their cells are eukaryotic, just like our cells. So it's hard to target their proteins and their cellular components and not have adverse reactions on our own cells due to evolutionary relatedness. And then do you remember there are antiprotozoan drugs which can kill protozoa. There are antihelminthic drugs that can kill helminths or worms. But then when we talked about antivirals, what did I want you to remember? Let's see if you can beat Wicket to the punch here. Wicket, do antivirals cure the patient usually? That's right, Wicket. No, antivirals mostly slow down the viral multiplication cycle, right? They slow down the viral spread. And it's up to your own body to cure and rid the infection, to clear the infection or not. Does that make sense? That's one key difference between antivirals as compared to the other antimicrobial chemotherapies we've discussed. And do you recall that resistance genes can be spread one of three ways to between bacteria through either conjugation through a sex pilus, transduction via bacteriophage, these viruses, or even transformation, which is the uptake of naked DNA across the cell wall. And then we talked about mechanisms of drug action. Remember, one example is to inactivate the drug. That's what penicillinase does. Another one is to decrease the permeability of the drug into the cell. Another one is to actively pump the drug out of the cell once it gets inside. Yet another is to cause an adaptation where there's a mutation changing the binding site of the drug. And lastly, forming an alternative metabolic pathway in the case of something like a sulfa drug that blocks a particular metabolic pathway. So concept check number 7, which of the following effects do antiviral drugs not have? Killing extracellular viruses, well, that's it right there, right? They can stop viral synthesis, they can inhibit viral maturation, they can block virus receptors, right? But they're not going to kill extracellular viruses. And again, antivirals typically do not cure, they mainly slow the spread. And do you recall that overprescription of antimicrobials is responsible for serving as a natural selection mechanism to spread antibiotic-resistant bacterium? And do you recall how superinfections work? When you take antibiotics, you can kill off your own microbiota, leaving that real estate open for pathogenic microbes to overgrow, and that's called superinfection. And this is responsible for gut diseases such as C. diff. Next, do you remember how we can determine drug susceptibilities in the lab? Remember that we can use the Kirby-Bauer disk diffusion test in order to determine which antibiotics are effective against the particular microbe that's causing the disease. We can remember, if a bacteria is susceptible to an antibiotic, then when you do the Kirby-Bauer test, you're going to have a large zone of inhibition. And do you remember that you measure the zone of inhibition in millimeters, and then you compare that to a chart to determine whether the microorganism is sensitive to the antibiotic, intermediate, or resistant to the antibiotic? The bigger the zone of inhibition is, the more sensitive the bacteria is to the antibiotic. And then do you remember that after we have selected antibiotics, then we can do an E-test diffusion test in order to determine the minimum inhibitory concentration that we need to prescribe to the patient. And that's it. Hopefully, that's a good review of all the important details. Please study everything. I do my best to remember those important exam questions and those important exam topics in the review. However, the best way to do well, as always, is to study all the material. I wish you well. I know you can do it. Study hard and do your best, and I will catch you guys for exam 3 material.
Dr. D, Dr. D, Dr. D. Dr. D, Dr. D, Dr. D. A Dr. D, Dr. D, Dr. D. Dr. D, Dr. D, Dr. D. A Dr. D, Dr. D, Dr. D. Dr. D, Dr. D, Dr. D.